This video is about taxonomic collection. So collection is the foremost and most difficult procedure of taxonomy. Collections can be either borrowed from museums or individuals or collected by the specialist himself. A thorough faunal survey of the geographical locality is needed before collection. Also, examination of previous collection is needed to get an idea about the fauna of the locality if a collection has already been conducted in that area. Field surveys face technical and political impediments. Many areas are accessible only through special permissions. Then taxonomic collections, there are various types. Types in the sense, many types are there. So biological specimens are preserved, plant or animal specimens along with the specimen documented, such as labels and notations. Various kinds of collections are dry collection. Specimens are preserved in a dry state. Dry collections are useful in following conditions. Rigidity. Some specimens can be preserved naturally, like starfish, or artificially with sufficient rigidity to help normal handling. The need for preserving specific characters. Drying is the best means to preserve natural colors. For example, butterfly, distinguishing features, skeletal features, or surface features. Almost any kind of container may be used for dry collection. However, tightly closed, impervious containers of metal, glass, or plastic should be free from moisture to avoid mold. Then wet collection. Some specimens are kept in liquid preservations to prevent disintegration. In general, ethanol and isopropanol mixed with water is the most widely used in wet collection. And most commonly, a mixture of 75% alcohol to 25% water is used. This water should be distilled to ensure a neutral pH, and the solution should be thoroughly mixed since alcohol and water do not mix easily by themselves. Then additives should be avoided. Specimens that are collected in this technique are usually soft body. This is done to minimize shriveling, allowing the identifying characters to be preserved as true to life as possible. Hard-bodied insects may also be preserved temporarily in alcohol before pinning. The liquid used for keeping the specimen should be carefully selected. For example, spiders preserve well in ethanol but tend to become flaccid in isopropyl. Then biological low temperature collection. Specimens are maintained at low temperature to preserve soft parts for various biochemical analysis by a technique called cryopreservation. Here, in biological constructs susceptible to damage caused by unregulated chemical kinetics are preserved by cooling to very low temperature, especially 80 degrees Celsius, using solid carbon dioxide or liquid nitrogen, minus 196 degrees Celsius using liquid nitrogen. At low enough temperatures, any enzymatic or chemical activity which might cause damage to the biological material in question is efficiently stopped. Cryopreservation methods seek to reach low temperature without causing additional damage caused by the formation of ice crystals during freezing. Specimens preserved at low temperature include some algae, protozoa, especially parasitic strains, viruses, cloned viral genome, bacteria, bacteriophage, plasmids, etc. Then animal tissue, dissected organs, muscles, cell lines, blood and blood components, whole blood, serum, plasma, antisera, semen, venom are also preserved in low temperature. Other samples include cloned probes, isolated proteins and nucleic acids, cell suspensions, etc. Whole organisms are preserved in a viable state. The largest organisms that can be preserved in a viable state are some insects. Then biological microscopic collection. Scientists preserve certain specimens as microscopic preparations to preserve whole or partial organisms for various kinds of microscopic examination. Some kinds of biochemical analysis including extraction of DNA. So types of microscopic collection: whole mounts, where an entire organism or structure is small enough or thin enough to be placed directly onto a microscopic slide, a small unicellular or multicellular organism, or a membrane that can be stretched thinly onto a slide. Squash preparations, where cells are intentionally squashed or crushed onto a slide to reveal their contents. Then smears, the specimen consists of cells suspended on a fluid like blood, semen, cerebrospinal fluid, or a culture of microorganisms. Sections, where specimens are supported in some way so that very thin slices can be cut from them, mounted on slides and stained. Sections are prepared using an instrument called a microtome. It is also common for microscopic collections to be ancillary to more traditional collections. Then purpose of scientific collection gives an idea about diverse species on Earth. Comparison of related specimens species gives an idea about how new species form. Documents biological diversity in a time of unprecedented environmental destruction, developing a baseline understanding of the effects of climate change and other environmental threats. Then collections in gene banks help in the study of how genes influence the structure and function of organisms. Genes and other molecular information can be retrieved even from specimens from the museum's historic collections, including fossil specimens. Tracks host and parasitic species responsible for infectious disease, determines the actual and potential impact of invasive pests on crop plants, discovers biomedicines, monitors the change in marine resources such as fisheries, then isotopes analysis in corals, and can reveal much about changing climates and oceanic currents.